Nobody's here.
Well, start. All the important people are, right? How you ladies doing? Everybody good?
Yep.
Yeah.
Time is. I'm going to record this one and try to get it on to Google Classroom. So if my brain explodes, you know what happened. Oh, Kristen, that is a cute picture. That's you when you were little?
Yeah.
Very nice.
Thank
What's that?
you.
Oh. You guys have any questions while we're waiting? No, we're all good. Is anybody else coming today after school? I think Daniel mentioned he was going to come pick up his egg. Anybody else? Hello?
I'm going tomorrow in the morning.
That's Denise.
Yeah.
Hey, I recognize voice that time. Okay. Yeah, I got the meetings and they'll let you in, I'm assuming, but otherwise I'll come get you and I'll put a picture of me when I was 2 up and.
Okay.
I'm coming tomorrow too, just so
Okay.
you know.
We had 1 ruptured egg, but I'll put a few more in vinegar. We're going to try to get at least a little bit of data from this. And I'll get some corn syrup. And remind me when you come, I got to get you a balance. I forgot to get you one, didn't I, Ari?
Yeah, but I'm going in tomorrow too.
Oh, you'll be in? All right, pick it up. And then if you guys want to do the cheek cell, I'm not requiring it. But if you've never seen your own cell, it's kind of cool. I think anyway. And somebody asked how long would it take? I'd say about 15 minutes. It's not a real long process if you want to and you never saw it. How'd you get all the way through high school with never seeing your cheek cell? How's that possible?
It's just possible.
No, I'm sorry. And even if it doesn't work tomorrow and you say, I still want to do it, but I can't do it tomorrow, come in whenever you want. All you got to do is scrape the inside of your cheek and you put the stain on and the DNA will get stained from the methylene blue. So it'll make a nice nucleus. If you look closely, you might be able to see some of the DNA strands, all 6 feet of DNA strands in every one of your cells. And in theory, I could take that 1 cheek cell of yours and make a whole new you, right? It's got all the instructions. I find that amazing.
That sounds great.
Do you want a whole new you? You could.
Yes, please.
Then you guys would be a little less busy than you are.
Yep.
You guys are crazy busy, you know that? Like I said, I compare you guys to me in high school and I was clueless, completely clueless. So I'm
Mr.
impressed.
T,
Yes.
what time should we go in tomorrow? Or to get the balance?
If you could do it before my meetings start or after my meetings, I think we go to like 1:30 or 1 o'clock. And I assume the meetings are going to start like at 8:30. But
Okay.
Listen, come when you want. I'll just get, because I'll be in my room anyway, and I'll just get you the stuff. I can miss a few minutes of the meeting. If you let me know when, that'd be great. Daniel.
Okay.
Yeah. Did you get my message about how long things would take?
Yeah, I think I'll be staying for the stuff you'll have to.
Yeah. So it won't take about 20 minutes if you want to look at it. And if you got, like I said, a jar, nobody's coming today in person, are they? I got 1 egg here ready to go if somebody wants that one. All right, we're going to go ahead and get started. Again, try to participate if you can today. If not everybody picks up an egg, it's okay. I'm hoping that between, I don't know, 6 or 7 of us, we get enough data that we can graph it. What we're trying to figure out is the solute concentration of the egg. So just to give you an idea, I took the vinegar and I used it as a acidic environment. It's got lots of hydrogen attacking the shell, and it destroys the shell but leaves the egg intact. What we're looking for is the right percentage of sucrose, that's the stuff we're going to stick in it, that will match the egg so that the egg doesn't gain mass or lose mass. And then if my wife really got into this, she had to add food coloring and make her egg bright blue. So we can do that too. All right. The idea is wherever there's more solute, there's going to be less water. That's where the water should go. All right. We're going to go ahead and get started. Remember, if you can, try to participate at least once today. I would really make me happy. Or rephrase. Again, I didn't learn my biology sitting there listening to it. If you can do that, you are going to be so much better off, right? Because I can catch your mistakes immediately. All right, anyway, we left off talking about this idea of, I'm not going to do this slide. I want to do the next one. Hold up. The difference between exergonic and endergonic, right? Which one are we gluing the vase together? Would that be exergonic or endergonic? When we're gluing the vase together. It's a million pieces on the floor. We're going to glue it together.
That's endergonic.
That is endergonic. We're going to have more energy at the end than when we started. If I break the vase, obviously, it's exergonic. Now, let's turn that into an organic molecule. What if I'm breaking down glucose by cellular respiration to get energy out of it? Would that be exergonic or would that be endergonic? Somebody other than Danielle this time, please.
Exer.
That would be exergonic. Energy is being released. In fact, so much energy is being released that I'm going to make like 600 ATPs from that one exergonic reaction. We'll talk more about that in detail later. On the other hand, what if I'm putting 2 amino acids together to make a protein? Would that be exergonic or endergonic? I'm gluing these proteins together now.
Is it endergonic?
Endergonic. Good job. Okay, so we're okay with those? Even if you get them wrong, it's good, right? Because then you know immediately whether you know what you're talking about or not. So we're never going to have cells at equilibrium. You don't need to write all this down, right? Cells are always going to be at an unequilibrium, right? They're not going to be equal because once they're equal, you can't do any more work, right? So when we look at these reactions, we're always going to couple what we call a catabolic reaction. That's also exergonic. Those mean the same thing, catabolic and exergonic. And we're always going to couple that with an endergonic or anabolic pathway. So for example, if you want to make amino acids, join amino acids to make a protein, that's endergonic. Where are you going to get the energy to do it? You're going to use an exergonic reaction, like the hydrolysis of ATP, the breaking down of ATP. All right, here's a great opportunity to say, Mr. T, so what you're saying is, and then try to rephrase it in your own words. Everybody understand that?
Yeah.
Good?
Mr. Chauvelin, can you repeat that again? I'm sorry.
Sure, no worries. So we're always going to couple an exergonic reaction with an endergonic reaction. Exergonic releases energy, and we're going to use that energy to make molecules join together, an endergonic reaction.
Wait, so what would be considered equilibrium in this case?
If a cell is dead, it'll reach equilibrium,
Oh,
right? And then you can't do any more work. I'll explain why in a second. That's one step ahead. Hey, Gael, how are you, buddy?
Sorry, yeah, good, and you?
Good, good. Hey, I was thinking, oh no, never mind. We already talked about that. Okay, moving on. So we just, we're reviewing a little bit. We're all going to participate. There's Perla. How you doing, Perla?
I'm doing good. Sorry, I was
No
Another
worries.
reading.
No worries. Good to see you. I'm glad you're here. So the major exergonic reaction of we're reviewing here a little bit. We're going backwards. By the way, I'm going to try to record this one and I'm going to try to put it on Google Classroom. Somebody suggested that that's what we do. So if that helps, whatever works, you tell me, okay? I'm going to have to figure out how to do it, but somebody will help me. My daughters will help me or something. Anyway, the major reaction we're going to use in our body is the hydrolysis of ATP. Now what that means is I add water and break a phosphate off. Yesterday we talked about phosphorylating molecules. If you give something a phosphate, it wants to react. And so poor Becca was here all by herself and I touched Becca on her shoulder and she creeped out, right? She became unstable and wanted to react, right? What did I do is I phosphorylated her shoulder. I gave her that phosphate and now she's unstable. So she immediately wanted to join to another molecule. What we use in our body as a currency is ATP. That's our cash. And the way we use it is by giving molecules phosphates. So whether or not we're doing chemical work, transport work like the sperm dance I showed you yesterday, or mechanical work, all of these are going to involve energy coupling. And the exergonic reaction we're going to use is the hydrolysis of ATP. Now, that was a tough thing to say, right? Did all those words make sense?
Use of exergon.
We use the hydrolysis of ATP as our energy source. We give things phosphates. We phosphorylate it. For the next, I don't know, 10 or 12 chapters, that's all you're going to hear about, is putting phosphates on things.
Exergon process to drive endergonic. Okay.
Anybody want to rephrase? Remember, our goal is everybody try to answer one question today or rephrase or just ask a question or clarify or whatever you want to do. This is not easy stuff, huh? So what ATP stands for, guys, is adenosine triphosphate. And adenosine has one ribose sugar in it. It's a pentagon because it's a 5-carbon sugar. It's got adenine, which if you'll notice there is a nitrogenous base, and it's got 3 phosphates. You're more than likely going to recognize ATP by those 3 phosphates. The one thing I want you to realize here is we're using a sugar and part of a DNA molecule to make this ATP. The body is very, very used to reusing molecules. It does this all the time. So adenine is going to be used to make DNA. It's also going to be used to make RNA. But here we're using it to make ATP. And on my next slide, I'll show you the structural formula of ATP. Kristen, you're still smiling there, so you look okay? Oh, wait a minute. That's not your real picture. So on your test, I'm going to put a picture of this. This was an old AP question. And they gave this molecule on the test, except they didn't tell you it was ATP. And they said, what could this molecule be? And they gave choices like this. Could it be ATP? Well, of course it could, right? Could it be part of DNA? Yeah, it's got phosphate and a sugar, and it's got a adenine. So it could be part of DNA as well. What it couldn't be is a protein. Why not? Somebody out there for gifted student of the day, tell me why would you say, hey, this can't be a protein? What doesn't it have? What do all proteins have on their ends?
Isn't it an amine group and a carboxyl group?
Gifted student, excellent, right? Doesn't have that. There's no way this could be a protein. Not to mention proteins don't have phosphate either. The better we get with these molecules, the better off we're going to be, huh? So that's good if you remembered that amine group and carboxyl group. All proteins are going to have them because all proteins are made of amino acids. Now, look at what we're doing here to this ATP. Somebody out there, here's one, you just got to describe it, right? in case you're worried about answering wrong questions. What's ATP giving off when it's exergonic? What's it going to release?
Energy.
Energy in the form of what, though? That is correct.
See water.
Well, you're adding water, yeah. And what's getting released? What do you see getting knocked off that I put on Becca's shoulder?
Phosphate.
Phosphate.
Yeah, the phosphate, right? And now it becomes ADP. Now the secret, another question off your test, and yeah, it's okay to write these down. The secret to ATP is the easily formed phosphates. They can go right back on to ADP, or they can go right back onto a molecule. They do this so quickly that they estimate, I don't know who came up with this, but they estimated that you can make 10 million ATP per second per cell. That's how fast these phosphates go back and forth. They are easily broken and easily formed. That's the magic behind ATP. Because of that, we're not going to be 20% efficient like our car. We're almost 40% efficient. There's a little bird, maybe this fall, we'll luck out and see him. He's called a black and white warbler. If he was a car, he eats those little seeds. If he was a car, when he travels down to South America, he would make 320,000 miles per gallon. How's that for a good car? He's very efficient. And his secret is ATPs. They're easily formed and they're easily broken. I pause for effect.
Okay, so
Anybody want to rephrase?
well, yeah, I'm coming up with like another question specifically on the energy coupling of ATP.
Yeah, coming to that next slide, sir. If you could hold that
Okay.
thought for 1 second.
Mr. Trevor, why
Yes.
is it that ATP becomes ADP and the P is not removed? Like from the description, I guess, of the molecule. Like, you know how it becomes from ATP, it becomes ADP when it loses a phosphate?
That is correct.
Why does the T become a D?
Oh, sorry, I didn't say that. You're right. The di just means two phosphates. Di refers to two in Greek, I think. And tri means three.
Okay, thank you.
See, that's me being teaching way too long and forgetting to say that. I'm glad you asked. So if you treat ATP like a charged battery, ADP would be like an uncharged battery.
Yeah, that makes sense. Thank you.
You're welcome. So here's what Daniel's going to ask. And again, before you look at this slide and just freak out and become totally intimidated, I want you to just look at the reactions that are occurring. This is a great example of energy coupling. And by the way, I left a space. I don't think you need to write all of this down, but in a minute, you can ask me what you need down in that space on page 2. And after I explain it, you may want to at least highlight part of this. This first reaction up there, is it endergonic or exergonic? When we're trying to add glutamatic acid to, we're trying to make glutamine. You think that's endergonic or exergonic? Somebody out there, tell me what they think. Yeah.
Endergonic?
It's
Ender.
endergonic, right? We're gaining energy. We're putting the vase together, huh? So this is going to take some energy. This never happens accidentally. This is not spontaneous. Look in picture 2, letter B. What exergonic reaction are we going to couple so that we make the endergonic reaction work? What are we breaking down in letter B so that we can get these two molecules to join together?
Ooh, okay, that was my question.
Yeah. What are we hydrolyzing? What are we breaking apart?
ATP.
ATP. It's a Native American home, by the way. ATP. ATP. Never mind. Bad joke.
Uh-huh.
Yeah. Now, where's Becca in letter B? Where did I put a phosphate on her shoulder? Do you see it?
Yep.
See that triangle? See the phosphate? I phosphorylated that
On the
molecule.
glucose.
Yeah, and it became unstable. Now it wants to react. And what am I left with? ADP, an uncharged battery. I'm phosphorylating that molecule so that it wants to react. All right, look at letter C. In order to join these molecules together, I needed at least 3.4 kilocalories per mole. Number or units are not important. When I break down ATP, it gives me negative 7.3 kilocalories per mole. If you do the math, that works. As long as we have enough energy to break down to join those two molecules with 3.4 kilocalories, it's going to work. We coupled an exergonic reaction, the breaking down of ATP, with an endergonic reaction, the joining of those two molecules. All right, I'm going to leave that up a minute, and let's see what we want to put down there on that blank. All right, you go away to college, yeah, you're going to have to really speak up. Your professor isn't going to let you do that, and but remember, you're paying for that education, so politely say, "Can you hold on a minute?" All right? You may not like it at first, but as long as you're polite, I think that's okay. You got to be assertive with your own education. So you don't even have to have those fancy names. If I'm going to join A and B together, it takes a positive delta G. Where am I going to get the energy to put those two molecules together? From breaking down ATP. What do you think? We should have questions about this. What's that?
Yeah, around at the bottom, I see that the net
Yes.
free energy is negative. So does that mean it lost?
Yeah, I always, like I said, I like the positive because I can wrap my head around it. The negative's a little harder. But the point is, when ATP breaks apart, it's not going back right away. It's released. That energy's gone. As long as the negative number is bigger than the positive number, the reaction will go. Right? We needed at least 3 kilocalories. I don't know how to respond to that. We need at least 3 kilocalories to get this reaction to go. And the breaking of ATP is 7. That's enough. Remember, the negative number means an exergonic reaction. We're breaking the vase. It's not going back. The positive number is the endergonic reaction. So as long as that positive number is smaller than negative 7.3, this reaction will go. Yes, sir.
Wait, okay. So the way I'm looking at C right now is that you need around 3 kilocalories per mole to join glutamic acid and ammonia, but the breakdown of ATP releases 7, which is more than 3. So that way it's possible.
That's it. Nice rephrasing.
Okay.
Yeah. And then we call that energy coupling. And in our body, that's how things work. If you're building anything, if anything's anabolic, it's ATP that's going to do the job. Other questions? All right, somebody who hasn't participated yet. What's going on in this slide? What are we doing in the letter A up there? What's going on? I don't need fancy names. Just tell me what you see going on. What got phosphorylated here?
Would it be the ATP?
Yeah, exactly, right? We knock the phosphate off ATP and we give it to this big purple molecule. What are we stuck with? ADP. We did work. Down here, this is how your muscles work, right? I took ATP and I phosphorylated your muscle. It becomes ADP, but the muscle contracted. You ever work out and get totally exhausted like I did when I was doing the trees, digging those trees, right? When you run out of all ATPs, your muscles can't move anymore? And then Lewis tells me, no, I'm not tired. I'm not sore. Is he on here, by the way? Makes me feel old. Right? He's making more ATPs than I am. What do you think? This is how our body works.
So you can run out of ATPs?
Well, it's
So you can run out of
for a little bit anyway, right? There are other sources of energy we'll get to later. Remember now, you have stored glycogen, so you can use glycogen. But yeah, you've been there, right? You tried to hold something up longer than you should have held it up, and you collapse. Well, you're out of ATPs, at least temporarily.
If we're doing movements, are we like breaking down ATPs?
Every second, 10 million per second per cell.
Oh, wow.
That's a miracle. If you need miracles, they're right here in your body. Every time you move, ATPs are being used.
Wait, so
Now, there's an
is it
issue.
possible-
What's that?
No, I was just gonna say, like, is it possible to be completely out of energy, like when you're not
It'd be tough. You'd be close to dead. Yeah. And again, we'll do the energy levels. You're going to use glycogen. You're going to use all the glucose in your blood. Then you're going to use glycogen. Then you're going to use fat. So there are ways to keep you going. Now, take a look here. It's a charged battery and uncharged battery. I constantly take ATP and use it as energy. I'm stuck with ADP. Does anybody see the problem? How are we going to get ADP back to ATP? Well, we need another exergonic reaction. That exergonic reaction is called cellular respiration. It's the second hardest chapter in the entire book, right? We'll get to it at chapter 9. But I just want to show you what cellular respiration looks like. Don't I have just a draw feature here? Oh yeah, here it is. Let me try this. So in order to put ATP together, I need this molecule. Sorry, it's going to be sloppy. Anybody know what I'm making?
Glucose.
I'm making glucose. I'm going to take glucose and I'm going to convert it to about 36 ATPs. That number is up for debate. So notice what I did. This time I'm coupling an exergonic reaction with an endergonic reaction. I'm using ADP and phosphate. That is my endergonic reaction. I'm gluing the vase together. And what I need is an energy source. I need glucose. Now, there's one other thing we need in this reaction. Anybody know what else we got to take in besides glucose? It's a gas.
carbon dioxide? No, oxygen.
It's oxygen. The reason you breathe is to make ATPs. ATPs per glucose molecule. So every time you eat food, you're going to convert them into ATPs. ATP is the cash of the cell. Glucose is like my paycheck. All right, we are, what time we, 12? I always forget this class. I was hoping to at least start enzymes a little bit. You guys, you got that in you? Let me hear some questions though first before we move on. Big deal, this molecule. Adenosine triphosphate stands for 3 phosphates. When you phosphorylate molecules, and you have to do that every time you move, I convert it into ADP, an uncharged battery. How do I get it back to ATP? I eat glucose. If I process cellular respiration, it makes 36,000 ATPs. Somebody's got a question out there for me, no?
So how do you change ADP back to ATP?
Nice.
You need
Go ahead.
Alright.
Keep talking.
You need oxygen or
You do need oxygen and you need glucose. Now, if you want to know more details, go to chapter 9, but I don't recommend doing that yet. That's the chapter that's like a badge of honor, getting through cellular respiration. But that's it in a nutshell, right? You take those two things in every day. If I take oxygen away from you, you will not make ATPs, you will die. If I take glucose away from you, you will also die. Why? Because you won't have any currency in the cell. No cash. What was that, Daniel?
Yeah, what exactly is it that combines the ADP and the P, like and the phosphate? Does a combination of oxygen and glucose, like?
Well, I'll give you the answer, but again, don't get nervous about this just yet. But it's a mechanical rotation of one of the transport proteins in the mitochondria. It literally is going to be like a turbine. And they've just figured this out recently, how it works. It's called ATP synthase, which is a nice name for it. It's a transport protein that converts the flow of protons to make ATP. Again, we don't need to know that yet. You'll see that chapter. It's very interesting. Anybody else? Okay, you guys got this graph on page 3? Maybe this is as far as we get today. Oh, by the way, I put up a practice test for this chapter. Remember, it's only one chapter, and it's only going to be like 14, 15 questions. So we won't do it. I think we had it scheduled for next, well, maybe even Monday. We're not going to be able to do that. I'll put up a new schedule next week. We'll just keep adjusting. We just don't want to fall too far behind. Does everybody see this graph?
yeah.
Okay, this is what I'm going to explain now. Now, the first thing you need to remember is that enzymes are
Protein.
proteins. They're made of what building blocks?
Amino acids.
Amino acids, right? So, and we know that they are in the right order because our DNA told us what order the amino acids should go in. Very good, guys. The tertiary structure of these enzymes is going to ultimately determine whether or not the shape works. When we looked at yeast, we looked at the enzyme called catalase. And if catalase broke down hydrogen peroxide, we knew that it did because it created bubbles. Now, the black line on this graph, the top one, is representing a reaction without enzymes. Notice that it is spontaneous. It's breaking the vase. We're going to knock the vase off the desk, and when we're done, we're going to release a bunch of free energy available to do work. But look at how much energy it takes to get to that point. There's like a little speed bump. So right here, this EA is called the activation energy. And without an enzyme, we have to use this big line and we have to add a bunch of energy to get there. It's going to take a long time and it's going to take a lot of energy. And in both cases, I'm going to try to erase that stuff back there. In both cases, it's going to take a long time and a lot of energy, which our body can't afford to do. Let me give you an example. How long does it take to digest your lunch or dinner? Well, about 24 hours. You'll convert it all into ATPs and carbon dioxides. That's because you got enzymes. Without enzymes, it would take an entire year to digest those food particles. You see why enzymes are so important? And here's the phrase you want to write down. I got some bullet points there under that graph. What enzymes do is lower the activation energy. It lowers the amount of energy needed for the reaction to start. It lowers the activation energy. Doesn't provide energy, it just lowers the activation energy. So we need a lot less energy to get this reaction to start. It's true that without the enzyme catalase, hydrogen peroxide will break down. but you'd be waiting there a long time. You'd get one bubble one day, another bubble another day. With enzymes, we're speeding up reactions by lowering the activation energy. Which line do you think is using an enzyme here? Which one is lowering the activity of the enzyme? The activation of the enzyme. Somebody out there, tell me red or black.
Wait, what was the question?
Question is, which line is representing the reaction with an enzyme? Which one is lowering the activation energy? The red line or the black line?
The red.
The red, yeah. The red is with the enzyme. You're going to get that one right on the test. And the black is representing the same reaction without an enzyme. Look at how much more energy has to be added in order for this reaction to occur. Now, I want you to look at the bottom of the graph where it talks about the products. On the test, I'm going to trick you, and I'm going to ask you, where is the energy of the level of products with the enzyme and without the enzyme? Notice where both lines end up. They do end up in the same spot. There's not more energy in the products. I don't care how much enzyme you add, you always end up in the same spot. If I'm breaking down glucose over here, right, I need to add a flame without an enzyme, but I end up right here at the same spot. There's only so many calories in glucose. Now, the idea of an enzyme is I don't have to add a lot of heat. Glucose will break down because I have an enzyme. That enzyme is lowering the amount of energy needed for this reaction to occur, but we end up in the same spot. So here's a tricky question one year they asked on the AP exam. They said, what if you triple the amount of enzyme in a reaction? What will that do to the amount of energy in the products? Anybody? Triple the amount of the enzyme, how much more energy will you get in the products?
Lower?
What's that? I didn't hear the answer.
I don't know.
Would it stay the same?
It's going to stay the same. It's going to speed up the reaction, right? It'll go faster, but the fact of the matter is, look where the black and the red line end up. They end up in the exact same spot. All enzymes do is speed up the reaction by lowering the amount of energy needed to start the reaction. But you see how you could fall trap and say, oh, triple the enzyme, then there's got to be more energy. No. It'll just go faster. Who added a lot more enzyme to their lab? Who was that? Is that you, Perla? Did you have a concentration of enzyme that was greater?
Oh yeah, that was us.
Oh, man, I remembered something. That's surprising. Good, right? And so you had a faster reaction, right? But the fact of the matter is you end up in the same spot, whether you had more enzyme or less enzyme. It just goes faster. Now, a couple no-dub points when we talk about enzymes. And yeah, the essay is going to be on enzymes for this one. And by the way, we're still writing the essays outside of class. You can use your book, your notes, because we're just not there yet. But we need to write more details. So tell me everything you know about enzymes if the essay involves enzymes. Enzymes are proteins. Enzymes lower the activation energy. Enzymes have an active site, and in a minute I'll show you what an allosteric site is. They speed up reactions, and are enzymes used up in the reaction? Yes or no? Enzymes used up?
No.
No, that's why we draw them on top of the arrows. Can I change slides, or are we still pondering on this one? I think I'm going to try to get one more slide in if we can. We won't be too far behind. Oh, not that one. Not that one. This is the one I wanted to show you. Okay. So let's introduce the enzyme. And by the way, it is okay to use drawings on an essay of the AP biology exam, but they can't be drawings by themselves. You have to explain them. But in this case, a nice little drawing wouldn't hurt, right? Notice that you got an enzyme there in purple, and you got the substrate. Now remember the substrate, you guys were confusing this when we did the lab. The substrate is what we're trying to break down. In our lab, it was hydrogen peroxide, and the enzyme was called catalase. The active site is where the substrate fits into the enzyme. And it only fits if those tertiary structures are in the right shape. How about letter B? Daniel, this was your part of the lab, correct?
Yeah.
Yeah. What would the competitive inhibitor, what did you add to your enzyme that kept the reaction from occurring?
That was alcohol.
Yeah, that was the alcohol, right? And in some cases, we're going to see what we call non-competitive inhibitors, where it's going to bind to a spot other than the active site. It's going to bind down here to this spot called the allosteric site. Somebody out there, easy question if you haven't participated yet today. What did it do to the active site when you bind it with a non-competitive inhibitor? Does that active site look normal to you, or did it change its shape? Anybody?
Did it change its shape?
Yes, right? So will the enzyme fit the substrate anymore?
No.
No, it's the wrong shape. That's what non-competitive inhibition would be. A lot of the poisons that we take are non-competitive inhibitors. They're altering our enzyme and keeping them from working. Now, I know, yes?
No, like I was saying, like when I was like doing some research on like inhibition.
Yeah.
When I learned about non-competitive inhibition, I originally thought it was that like if the inhibitor binds to another part, then the enzyme must also direct its attention there, which means that the substrate doesn't get the full attention. But now this clears things up.
Yeah. Yeah, and you see where I wanted you to go one more thing in that lab by adding more of the substrate, right? Then we could have proved it was a competitive inhibitor. You know what I mean?
Mm-hmm.
I wasn't sure if it was a competitive inhibitor or not. That make sense?
Yeah.
All right, last thing here. I'm going to explain if you want. I feel terrible that all you guys got B's now because you have a 92 instead of a 93. That is such a steep grading scale. You guys are very impressed. Anyway, I'll give you some extra credit. Watch the toothpick biology bite where I blindfold my wife and she becomes the enzyme. And if you can do your own video, less than 1 minute long, grab somebody, blindfold them, and then explain it in a 1 minute video about competitive inhibitor and the active site and the non-competitive inhibitor, I'll give you extra credit. I don't want more than a minute, right? You got toothpicks in your bag, just grab your brother or sister or mother, whoever, and just do a quick demonstration with the toothpicks. You'll notice I gave you different colored toothpicks, right? So those would be the competitive inhibitors. Anyway, if you want to do that, that'll help your grades some. Questions?
When will that be due?
At least before we take the test on the enzymes, huh? Again, I'm not looking for anything elaborate. I'm talking 1 minute.
Okay.
Right? In other words, you know, go over in your head what enzymes do, use the toothpicks, grab your mom or your sister and just do a quick demo. Okay? And it won't be worth millions of points, but again, it'll help. And people ask, hey, when do you put all the extra credit in? At the end of the semester, if you're between grades and you've done extra credit, that's how it'll boost you over, right? So it's not anything, I keep telling you guys to be quantifiable. Well, it's not really, my extra credit isn't quantifiable, but boy, it does make it easier to say, hey, this guy's come in for labs, he's done this, he made a video, blah, blah, blah. then I rest well at night, giving you the A instead of the B plus. You know what I mean? Okay, but again, this should not take long. This should take a couple of minutes tops.
Okay.
Okay.
Yeah.
All right. Thanks, guys. Did a great job today. Come get your egg.
Trevaline, I have not a question, but just to make it clear. So it doesn't matter whether it's competitive or non-competitive, but the inhibitor still like affects the enzyme, I guess you could say, and then the substrate and how it reacts with it.
Yes, they both affect it. However, the one thing about a competitive inhibitor is if I add a lot more substrate, if I add a lot more toothpick, I can outcompete a competitive inhibitor. Generally speaking, a non-competitive inhibitor alters the shape and it won't help.
Yeah. Okay. Thank you.
You're welcome. Thanks, guys.
Thank you.
Okay, bye-bye.
Mr. Treveline, what time do we go tomorrow morning?
You can. I'll be here all day because I got meetings all day. If you want to try to catch me before meetings and it's not, yeah, I'd say like 8 o'clock ish.
Okay, yeah, I can go around 8 because then later on I have my college counseling.
Okay.
Yeah.
That's not too early for you. If it's a matter you
No.
get sleep, just come later. I'll interrupt my meetings.
No, it's okay. I'll come at 8.
Okay. When anybody gets here, I'm gonna interrupt and just do the lab. So
okay.
So if you
Thank
come
you.
later, that's okay. All right.
Yeah.
And text me if you're having trouble getting in or whatever.
Okay, thank you so much.
Sure. Bye-bye.
Bye.
Hey guys, how are you? You guys need a lab, go grab that lab. Oh, let's see. You were right last year. I probably imagine my Titans could be the screen right there. The screen right here. I'm gonna get there. I do. I give you 7. Okay, you guys want to lab? What's going on? You're confused, you okay? Let's finish. Hey, I'm your teacher now. If somebody's bugging you, tell me I'll beat them up. Oh, very good. Thank you. No worries. So today, you're going to make a food lab out of my compost. My compost is an ecosystem. And the more creatures you find, the better your food web will be. Okay? If you want gloves, there are garden gloves up here. You can use those. The ones that are out here, hold on. These ones, don't use these or that one. Any of those are okay.
I don't know. Maybe it was on the
Right here. Oh, you don't think so? Find out. Oh, please. No. You guys each need a tray. How we doing? Okay. Yeah. Everybody looks sad. This is gonna be fun. Now, if you don't like it, then just find your 5 organisms and call it a day. and we'll have time to feed. In fact, you guys are the last class, so you can feed every worm we find. We'll feed them to the turtles at the end. Okay. That sounds good. Is anybody on Zoom? Grab one of those trays, in the jar.
Isaac.
Martha should be on Zoom. Isaac and Martha. Isaac and Martha, you there? I'm gonna start over. Okay, you guys read the lab and I'll be right with you. Let me see if I can find Isaac and Martha.